John's lecture for today will be delivered by Dr. Carlo Colantuoni, an assistant professor of neurology and neuroscience at Johns Hopkins and adjunct professor at the Institute for Genome Sciences at the University of Maryland. Dr. Colantuoni pursued undergraduate studies at Princeton before his neuroscience PhD at Johns Hopkins and postdoctoral fellowships at both the NIMH with Dr. Joel Kleinman and at the Hopkins Bloomberg School with Dr. Scott Seeger. Dr. Colantuoni's group works to elucidate molecular mechanisms in human cell biology by developing and applying novel data decomposition tools to curated collections of multi-omics data sets focused on particular cell and disease contexts. Their approach interrogates both in vivo tissues and in vitro stem cell systems to gain deeper understandings of human functional genomics. He's worked extensively as co-investigator of creating the Neuroscience Multi-Omic Archive, or NEMO archive, and the companion NEMO Analytics platform. He leads the neocortical development data exploration environment in this and implements projection tools for delineating shared molecular dynamics across biological systems. And relatedly, his talk today is entitled Harnessing Public Multi-Omics Data for Discovery in Your Neuroscience Research. Thank you for being here, Dr. Colantuoni. Please go ahead and share your slides.
Really appreciate it. Okay, are these visible to everyone now?
Yes.
Yes, they look good.
Great. Thanks so much again for the introduction and the opportunity to share my work. I strive in everything I do in the quest for my own discoveries to make the things I use to make those discoveries as available as possible to others. So today I'll actually be focusing less on the discoveries I have made and more on the elements and the tools and resources I've developed to make them. Because at this point they've reached a level of maturity that I hope immediately they can be of use to many of you in your own work, whether that means that you're already into multi-omics and you have an RNA sequencing data set that you want to get more out of, or if you don't have any multi-omics data, but the cellular or disease context that you study could be presented to you, or you could get into it more via this kind of data, even if you don't have bioinformatics skills. So, and please reach out to me after if any of this seems like it could be useful in your work. Like I said, we've spent years developing these tools and now they're kind of ready to fly and be used by people. Okay. So it really is about how we can empower research. So the only disclosure I have is that part of my research is supported by a local biotech company called Red Abbey Labs that's into finding new therapeutics for neuroinflammatory conditions. So here's the objective that perhaps we can direct ourselves toward in our conversation today. And actually, please do interrupt me if you write during the presentation if anything isn't clear, because there's a lot of different things in the presentation that I will try to explain, but sometimes glance over. So, and I know the audience is diverse and the talk is quite diverse in the things it covers, so don't hesitate to interrupt. But the goal is, through the things I show you today, that you'll actually be able to go out and grab data and look at data that can inform your own research, whether or not you or your group has bioinformatics or coding expertise on hand. Because it's really the underlying motivation for this is that any field in neurology and neuroscience, or really biomedicine today, there are literally millions, if not billions of dollars worth of data out there relevant to what you study. But getting that in your hands and using it to understand what you're looking at is not at all easy. So data is placed in public repositories, but it is not at all accessible to people who don't code for a living. So really the underlying motivation here is to get data into the hands of researchers globally. And today we're going to be talking about several neurodevelopmental and neuroinflammatory contexts that I hope are relevant to more people's work in this audience immediately. so that you can start using some of this incredibly valuable data we're generating. The scientific community. Okay, so I'm going to outline the general approach that we take to looking at scientific problems. The way I contextualize it is trying to find conserved molecular dynamics, whether that's conserved across species, conserved across diseases where common mechanisms are used, or across cell types where two different cells are actually expressing the same module of genes in a particular response. So conserved molecular dynamics across cell and disease contexts. So step one is get the data. So in several different contexts, we've been working for years now collecting multi-omic data. Most of that is transcriptional because the RNA world is what's sort of the technology is there to study incredible amounts of RNA across tissues and single cells. So in any one disease realm, be it neuroinflammatory or neurodegenerative disorder, there are data sets that are relevant across mammalian species, if not from even additional species. So there's all sorts of things to get there. There's model systems going from in vivo work in animals to in vitro work in a dish or even organoids, to human tissues, of course. And levels of analysis, you can get bulk tissue and see what's in it. There's sorted cell types. There's microdissection with laser microdissection tools. And then there's tons of single cell analyses now in nearly every tissue you can imagine. So again, this data is out there. It is incredibly complex, but it is incredibly useful. So how can we get more of it together in one place in all of our hands to use? There's, like I said, most of what we'll be looking at today is RNA sequencing, but there are tons of new data sets out there or new data types out there that are also of use. So everything I show today will be on the RNA world, but keep in mind that any data set that can be summarized at the gene level could be used in the exact same way. You can look at ATAC-seq, which shows open chromatin, if you quantify it at genes and then integrate it into the entire system I'll be sharing today. And the realms I've been most active in so far are pluripotent stem cell models, which are most directly relevant to developmental things, but as the stem cell community advances at a lightning pace today, nearly every organ in the human body is being studied in a three-dimensional pluripotent stem cell system today. So what you're studying in patients is probably in a dish somewhere, and people are sequencing it, and the data could be useful to you. Perhaps the realm I've looked at most is neocortical development, and I'm moving really into neuroinflammation and neurodegeneration that I know many of you in the audience study and will be interested in studying. So again, hopefully after today, immediately, there'll be some things you can go visit online that are already public, already functional, that can help you contextualize what you're looking at in a system, because no one of us as a group or an individual can look at everything that we're studying, but the data is out there to help us really look more deeply at what's there. So step one is get the data. What is step two? So I call it decomposition and projection. So now we're getting into some math. I won't talk about math today, but the goal of this second step is to take these data sets we've collected, and we can think of each one of these data sets as a big Excel spreadsheet where the rows are genes and the columns are samples. So it could be, an LPS experiment in vitro where you have some cells that were treated or untreated in those columns. The columns could be individual cells of the human brain where the individual cells were sequenced. And then always we have all the rows that are the genes. So any one data set we're conceptualizing today, think of it as a matrix or a spreadsheet. And we collect hundreds of these data sets and integrate specific ones that are relevant to each other to find things that are robust observations and that are common across these different experiments. So decomposition would be something like taking the differentially expressed genes from one experiment, or maybe even a principal component that gives you a weight for every gene, but some gene signature that is relevant, and it could even be, let's say, table 2 from a paper you're interested in that lists microglial or genes that are activated in microglial activation, and you want to know something about that gene packet. So we're decomposing data sets to get gene signatures that are biologically meaningful. And then the second step of that is projection, to go looking at other data sets with that gene signature. And the references I have up here are for the more mathematical references and papers we put out in recent years. I have long-standing collaborations with a lot of math friends in the School of Public Health, as well as in math and applied stats at the Homewood campus, because we talk about these many, many hundreds of data sets, and each one of those can have, somewhere tens of thousands of rows when we're looking at genes, and today even up to millions of columns when we're looking at single cells in a single data set, and we're looking at hundreds of these. So the math matrix, or the matrix math techniques to look at these data sets and to decompose and pull out relevant pieces is really a huge area of active work, but I'm not going to talk about those techniques. Please do reach out if you'd like to learn more about them, but I'll just show you how they're used today. So then the third step is get all these data sets and signatures from decomposition into a place where not only one or two bioinformaticians can use them, but where the entire world, anybody can use them with, say, a minimum of biological knowledge. So nemoanalytics.org is the website that for years we've been developing with colleagues at the University of Maryland. It is absolutely up and public and functional. So you can go explore there. There's so much there that it's a little bit hard to just walk in the front door and use it. There's some help there at that site, but also my own website which I list here too, which is just my name.org, has sort of a guided tour to the data resources that I will be presenting today. So feel free to use those, and I'll definitely send out links to the same email list as the Grand Rounds email went out to, so that anything I present today, it'll be one click away if you're interested in using it. So the picture at right is a screen capture from one of these data environments in the NEMO Analytics. It's from that neocortical one that I said is quite developed. We have about 200 papers worth of data, which turns out to be, in this day and age, it's about 30 or 40 million samples across hundreds of data sets, where each data set we have in a little box just like this. And if you're not used to looking at this kind of data, I'll take a second now to kind of walk through what these pictures are. So each one of these boxes is a different single cell RNA sequencing experiment, where each dot on this plot, for instance, is a single cell from the developing human brain. The picture to the right gives you a key to what cell types are on this map. So back here, in what looks to be sort of the tail of this swimming whale, are the progenitor cells. These are the neural progenitors of the neocortex. One side of the whale tail is here. The second side are those progenitors that are actively dividing. And just a sort of a flashback to neural development, if some of us haven't thought about it in a while, those progenitors, the radioglial cells, go through an intermediate progenitor state before turning into neurons, which is all the orange here. So this is a snapshot of the developing human brain, where each dot is a single cell, and their gene expression profile. So we have 30,000 gene expression measures from each one of those dots, each one of those cells. And if you have similar expression values, you're close together on this image. And if you have very dissimilar expression values, those cells are very far apart. So this is a particular gene that marks that intermediate progenitor cell state that is sort of the transition from a radioglial progenitor to a neuron in the human brain. And if you go online, you can just type in any gene name you're interested in, and it will paint its expression across all of the hundreds of data sets we have there. That's sort of the level one, easy access. If you have any tissue you're interested in or any specific gene, there are huge data collections that you can just look at here. And this is really just about getting data together. This is years of data here. You can see on the side there, some of these are from 2015 when single cell sequencing just started. We have all the way up to the most recent ones. And that's what I mean by kind of level one. You can go here, type in a gene name, and look at relevant data sets. Where is this gene expressed? What cells is it in? Is it higher in cells in a particular area of the brain, et cetera? And there's unbelievable data sets out there, from these single cell data sets that have millions of cells to slices of brain that aren't stained for one protein, but for which we have 30,000 genes worth of information on every pixel of an image of a brain slice. So that the data sets are only becoming more amazing and more useful. So again, this is really about getting it in our hands. So I'm going to do a flash to a live window to show you what this looks like in Nemo, which is kind of always a no-no in a presentation because it'll always go wrong, but we'll give it a shot. So this is the live web page where the screenshot came from. And as you can see here, I just typed in a name of a gene that I was interested in. Again, this is a marker of those intermediate progenitor cells. And if I scroll down on the page, every one of these boxes is a whole matrix of data from another paper and showing us on the right-hand side, the map of what they charted, the cell types, and then on the left, the expression of your gene of interest. And I'm not going to go through these, but I'll just scroll down a little bit to give you an idea of the immense data sets that are out there. So most of these at the top have been single cell. There's some of these incredible data sets that if you click and zoom in on, each one of these dots is actually a laser captured region of the developing human brain that was then subjected to gene expression analysis. So here you can see in these particular regions of the brain, the developing brain, this is where these intermediate progenitors hang out in the germinal zones. Where the actual neurons are, it's much lower here in green. But this is just to give you an idea of the vast extent of data sets that are out there. On this particular page that is relevant to in vivo human neocortical neurogenesis, we have about 50 or 60 studies, including all sorts of different modalities. So again, if you're studying a microglial gene or a neuronal gene and you want to see where and when it is expressed, there's incredible resources to do this. So flashing back to my slides now, and I'll do a little bit of this as I explain something. I'll flash to the live website to give you an idea of what it looks like there. And again, I'll send around links to go through all of this. Okay, so now I'm going to roll forward and basically give you 3 examples, well, depending on timing, 2 or 3 examples of this process, getting data together, doing some decomposition and projection to look across different data sets and start to really leverage the diversity and depth of data that is out there. So I'm going to start in neuroinflammation. So this is a data set produced some time ago. It is actually from 2012, which in today's years seems like a lifetime ago, but it's a super valuable data set still today. It was from the late Ben Barris's group, where they did peripheral injection of LPS mimicking a bacterial infection, but then grabbing astrocytes from the CNS, purifying them, and then sequencing their RNA to see what innate immune response or any response is going on in central astrocytes in response to a peripheral infection or immune response on the host part. So the pink and red are the LPS experiment. They also had a stroke experiment in here, occlusion of the middle cerebral artery. But what I did was grabbed the top genes that were different between the control and the LPS group. So that's actually what's on the y-axis here, is the composite or summed up expression of the top 25 genes that went up upon peripheral LPS challenge, but expressed in CNS astrocytes. So this gene list is interesting in itself, but once we put this gene list or this gene signature into the NEMO system, we can then not only look at this signature in the data set that it came from, but in any other data set that is relevant to our own thinking. So get rid of these for a second. So that's decomposition, maybe the simplest form of decomposition, grabbing a small list of genes from a huge data set. So we're grabbing a list of 25 genes here, and then we're going to look at the expression of those 25 genes in some other contexts. So here's another experiment from Paul Teaser's group, much more recent, just in this past year, with both bulk and single cell RNA sequencing. This is now in the mouse, again, as the Barris experiment was. And they're looking at cytokine treatment of CNS purified astrocytes. So similar, but not exactly akin to the LPS experiment. But what we can see here is that this packet of 25 genes going from control to different cytokine treatments is also turned on. So it's a common thing if a astrocyte is exposed to tumor necrosis factor alpha and a few other cytokines here, or if you give LPS and let the immune system do that sort of activation for you. And that's evident here at the bulk level and here at the single cell level, where this group themselves classified reactive versus non-reactive astrocytes. And this is actually purifying those astrocytes, putting them in vitro, and then applying the cytokines. So these are in orange untreated, and then cytokine treated in blue. And you can see again in this map where all of this orange comes up, the cells, i.e. the sorted astrocytes from the CNS, are turning on the same packet of genes that the mouse astrocytes turned on when they saw LPS peripherally. So again, this is a way to connect common molecular dynamics across very different experiments. So now we're just going to walk through a couple examples. Here's another example, again, looking at the same packet of 25 genes, now in single cell sequencing, not of astrocytes in particular, but all of the CNS cell types. So there is a small packet of cells expressing this signature at high levels. There was no challenge in this experiment. So there's not a huge number of these reactive cells, but the cells that are lighting up here correspond to some cells in this green cluster. And if we look down here to the green, these are actually what they, in their own study, classified as microglial cells that are disease-associated microglia. Now they say damn like because there is no disease here. This was a developmental study. But the point is here that we can see something that we would not be able to otherwise see. The astrocytic reaction to LPS is actually turning on a common set of genes that a certain small population of microglial cells are turning on here in this developmental context. So then this is maybe not a surprise for many of you who think about the innate immune system, where a lot of astrocytic and microglial expression modules are in common. But this is a way to actually quantify and identify them across systems. And just one more example here. This is some single cell RNA sequencing from severe COVID infection. There's a clear cluster of cells over here that is expressing the astrocyte reactive 25 gene signature we have over here. And those actually turn out to be microglia. They're actually expressing this packet of 25 genes at much higher levels than astrocytes. The astrocytes here are in red. And if we zoom in a little bit on this, you can see there's some pretty dark dots within there, but as a population, for sure, the microglial cells are really expressing this signature at higher levels than astrocytes. And again, this is just examples of capturing something that you're interested in. It could be, 5 genes that are central to some particular cell context you're interested in, and then you can use those signatures to go explore more broadly. And so again, I am going to flash to the live site here. This is the neocortical. This is now a neuroinflammatory collection of data sets. So each data set in a box here is going to be somehow related to neuroinflammation. And instead of having a single gene be shown, we have this reactive astrocyte LPS signature loaded up. So we uploaded this signature to the same environment where all the data is. And I'm not going to go through these examples in depth because I just showed you a bunch. and the ones we just looked at are sprinkled in here. But as you can see, and this is that COVID example, there are many, many data sets. This is some cerebellar data from children who died under a variety of neuroinflammatory conditions. And again, it's the microglia that really show this astrocytic signature. But there's just a wealth of data across the world in different contexts. There's some HIV data, more LPS data. This is MS from microdissected lesions in the human brain. This is from HIV infection. And again, what we're looking at, and all the three examples I showed you, are different data sets from very diverse contexts that we can now see whatever gene signature we're interested in. Okay. So now I'm going to walk through one or two more examples. And as I do that, we're going to get a little more fancy and complicated about how we capture signatures, that decomposition and projection step. The decomposition is capturing signatures, and the projection is looking at those signatures in a next data set. So I will only touch on the biological discovery from this paper that we published just this year, but it sort of breaks my heart not to get deeper into it. It's some of the most exciting work I've done. But what I really want to tell you today is how we did it, rather than all the details of what we did, but I'll get into a little bit. So these two data sets are very different. One is bulk RNA sequencing here on the left. So every dot here is a dish of pluripotent cells from a different human. And they had just over 100 donors. And they didn't only sequence the cells from each donor once. They actually had multiple pluripotent stem cell lines from multiple individuals. And what this is the first principal component, or the biggest dimension of change across all of those pluripotent stem cells. And they're ranked by that first principal component that's plotted on the Y. The X is the donor, just ranked by how high it was on the PC1. So this means that any dots in a vertical line come from the same donor. For instance, these that are circled in blue are 3 different pluripotent stem cell lines from the same donor because they're in the same vertical line. And I've highlighted the one in blue because one of its pluripotent stem cell lines was acting very different than the other 2. And you can see that's kind of rare because there's not a lot of variation in this direction. Most times when you study the same person's human pluripotent stem cells, they look quite like themselves, which is good for the system. It gives us sort of some confidence in what variation we're seeing. But as you can see, the spread across the y-axis is much bigger. These person cells and these person cells are at total opposite ends of this spectrum of variation that exists across human pluripotent stem cells. So what is it that is different across these hundreds of people's pluripotent stem cells? Well, the color of these dots is scaled to reflect the expression of retinoic acid response genes. So retinoic acid is a morphogen that controls the development of our cells and tissues from the earliest steps. So it turns out, and it controls many dimensions of that we won't get into today, but so what it turns out is that PC1, the biggest variation across people's human pluripotent stem cells in a dish, is how highly they express these genes. So as you can see, all of these people's cells here are expressing high levels of retinoic acid, or retinoic acid response genes. And people down here are all in red. In fact, all of this down here is quite flat. These are people who are a little lower on that spectrum. Their pluripotent stem cells are expressing the retinoic acid response genes at lower levels. Super interesting, but what does this mean? So here now we are going, that's our decomposition, PC1. The main dimension of variation across human pluripotent stem cells in quite a large population. So what are we going to do with this signature to understand what it is? That's where this second data set comes in. This is in the mouse. It's in very early development. I believe this is E6.5 to 8.5. So those are 2 days in the 20 days gestation of a mouse. And so every dot on this plot is a single cell that single cell RNA sequencing was done on. There's actually tons of primate and human data at this level now, but this is one of the earliest and most detailed atlases of early development. And so what the color scale is that's on this map is going from low PC1 to high PC1. So the purple here, that is the pluripotent epiblast. So that is the stage of very short period of time after conception in mammals where we don't, prior to gastrulation, where the cell types of the body have not yet emerged, and that ball of cells, or in a mouse, it's more, they call it an egg cylinder, that ball of cells has the potential to produce all the cell types of the new animal. So the pluripotent epiblast is very low in PC1. And as this is a map of going toward the CNS, going toward endoderm, or going toward mesoderm here. So what we can see here is the spectrum of variation that we captured by just looking at human pluripotent stem cells before they differentiate. The difference between human pluripotent stem cells is actually along the axis of the emerging lineages in early mammalian development. So different people's cells in a dish are actually biased toward these different lineages. And of course, anybody who has created, or any, let's say, human genome that has created a human body can produce all of these lineages. So we don't really know what it means yet that one person's pluripotent stem cells are slightly tilted toward producing one or another lineage, but we know that is the case. And we can, in further analysis in this study, we actually track down transcriptomic signatures that are specific to individual humans and all the cells of those individual humans. And those transcriptomic signatures control these biases. And you can see them in people's pluripotent stem cells, and you can see them in their tissue, their blood cells or their skin cells or their brain cells throughout the decades of their life. So it's a consistent, stable molecular trait that's stable across our lifetime and that impacts the behavior of our cells. So we think this is actually a great step toward opening up avenues in precision medicine. And we got it by analyzing pluripotent stem cells in a dish, understanding variation across humans, grabbing a principal component in this case, but we could think about many different ways for grabbing gene signatures from here that might be interesting, and then understanding them through totally different data sets in other contexts, in this case, a very early development. So just like before, I'm going to jump to the live website one more time to show you this data. So here is that data I just showed you, but in a live version online at the Nemo Analytics website. So here we have NextGen PC1, and NextGen was the consortium that gave rise to those hundreds of pluripotent stem cells. So this is the PC1 right here. It's just displayed differently, but this is the version in the online system. Here we're looking at the level of that PC1 across all the donors. It's colored by retinoic acid, just like in the other view, and we're projecting this PC1. So all of the data sets below here are additional data sets to understand what this PC1 is. These individual frames are actually the same data set that we see here, but broken into 6-hour intervals across those 2 days of development, so that you can actually watch what cells where are expressing this signature as we go from E6.5 to E7.5 to E7 and so on. These are temporal steps. And then here at the end is the whole map, again, showing pluripotent stem cells at one end and the mesendodermal lineages emergent at the other. And so this is a similar study in a primate, where here again is the pluripotent epiblast that is very low in this signature. And as lineages emerge, in the mesendodermal lineages emerge, they turn it on. That's what the darker orange is there. Okay. Okay. So we looked at a neuroinflammatory example with a quick grabbing of 25 genes from a differential expression analysis. Here we're looking at super early development, some of the earliest steps of lineage emergence, and we grabbed a signature of human variation by taking a principal component across hundreds of different people's cells. And I think I have time, yes, we're looking pretty good, to give you one last example of how this approach works, and getting a little more complicated as we have been each time. So we started with one data set, grabbing 25 genes in that neuroinflammatory example. In the pluripotent stem cell example, we did principal component analysis of a library of human RNA-seq, but again, one matrix or one data set. Here, we are going to leverage some of the fancy matrix math that I mentioned earlier on that I've developed with colleagues to harness signals not in one data set, but in three. What are the data sets? This is all in vivo data from neocortical neurogenesis across mouse, macaque, and human. So mid-gestational neurogenesis is when the vast majority of the neurons that we think with in our neocortex during our whole life are born. A couple weeks right in the middle of gestation. And then we live and think with those same cells our whole life. So this is there's a 6-day period in the mouse, again, right around the middle to late progress in a 21-day gestation. In the monkey, it takes about 160 days of gestation. This is again right in the middle there, between day 50 and 100. And then in the human, mid-gestation is right around week 16 to 24. So each one of these pictures is again a single cell RNA sequencing experiment where each dot is a cell that was captured and all its RNA sequenced. So just like we took a principal component of one matrix to look at that human pluripotent stem cell variation, here we are going to look at components, but they're not just going to be from one data set. We've developed algorithms that can look for dimensions that are shared across data sets. So what this is a picture of 4 sequential dimensions that were defined in this way. They sort of go from the bottom of each picture and move right on up. And this is the stem cells, intermediate progenitors, new neurons, and then neurons. And I'll walk through that a little bit slower, but I'd like to use the animation to show how it's flowing through each of these systems in a very similar way, even though the dynamics are, or the shapes of these cell clusters are a little bit different. So here is the radial glia, or the neural stem cell signature. I have down here to remember to mention that genes in this signature map onto risk for microcephaly or changes in brain structure. Here, right after the radioglial signature, we have an intermediate progenitor signature, where progenitors are now transitioning into the postmitotic neuronal state. Right after that is a transcriptional signature that is very specific to newborn neurons. This maps back to risk for autism. Tons of autism risk genes are in this packet. Now, this is a particularly interesting one because it's so transient. This disappears within days in a mouse and within a large number of days in the monkeys and maybe within a week or two in the human. It is just expressed as a cell is becoming a neuron. And as you can see, so this is sort of where the IPCs ended, and this is where the neurons are. So this nascent neuron signature right in here is again being turned down at the end of each one of these trajectories. And what is being turned on there is the maturing neuronal signature. So you can kind of see how we're leveraging multiple data sets now to have, behind the pictures we just looked at is a very quantitative description, gene by gene, of which genes are involved in which of these signatures. And this is preserved across the mammalian lineage. Mice, which are almost at the bottom of that tree, show a beautiful progression, just like monkeys and humans do here. So now this is our decomposition step. We now have detailed transcriptomic signatures that are conserved across mammalian neurogenesis. Now comes the fun part, projection. We can look at these signatures in new data sets that have other pieces of information. For instance, these are all single cell studies, which is incredibly cool, high resolution, cell type specific information. But formally speaking, we have no idea how these cells were arranged physically in the tissue before we sequence them. Before this experiment happens, they're just all torn apart and separated and then sequenced separately. Okay, so how do we do that? Okay, this is now a redisplay of the data we just looked at. There's nothing new here, except I've rearranged it a little to get ready to look at lots of data. We now have our mouse, monkey, and human data sets here in each row, and the 4 signatures we just looked at separately of the neural stem cells, intermediate progenitors, newborn neurons, and maturing neurons are in our columns in the same data set that we derived them from. So I'm showing you this just to orient you basically to this next slide, which is our super fun projection step. So like I said, why do we project? Because we want to know more about the thing we just found. So this is, again, maybe a flashback to some neurodevelopment that we might not have seen in a while. But like I said, the signatures were captured from the 3 single cell data sets that have incredible molecular resolution, but have no tissue or spatial resolution to them. We don't know where those cells were in relation to one another when they were captured and sequenced. But what we're looking at here now, the same 4 signatures are again in the 4 columns, but now each row is a brand new data set that was not involved in getting the signatures. It's just a new data set we're going to look at to understand the signatures. So this is an incredible data set produced a couple years ago, one of the biggest spatial transcriptomic data sets in the developing brain that's ever been produced, from Chen et al. In China. So it looks like a immunohistochemistry or an in situ hybridization section, a section of brain that was stained for one protein or for one message. But in fact, what's behind those images, for every pixel in that image, we have the expression of every gene in the genome. So what enables us to do is to take those genome-wide signatures for radioglial cells, intermediate progenitor cells, and the 2 neuronal signatures, and explore where they are in what cells and exactly where in the developing brain. So this is E16.5 in the developing mouse brain. So that's right in the middle of the mid-gestational period that I mentioned, where all this neurogenesis is happening. So now when we project these different cell type specific signatures, we can actually see physically where they are, even though the data we got the signatures from had no spatial data in it whatsoever. So where are the radioglial cells, the neural stem cells of the telencephalon? Well, they line the ventricle of the entire CNS, from our telencephalon all the way back to our spinal cord. And where is that here? That's everywhere where you see dark cells here. So this LV is the lateral ventricle, and as you can see all the way around the lateral ventricle, there's these layers of cells, the radioglial cells that are the neural stem cells of the telencephalon. What we also find out here is that this is not a telencephalic specific neural stem cell signature. It is lighting up the cells that line the ventricle across the entire CNS. So we've captured not only a species conserved, but a brain region conserved signature of mammalian neural stem cells and so you can find a neural stem cell anywhere in any tissue by shining this signature through it. The IPCs, the intermediate progenitors, are produced by those stem cells that lie on the ventricle, and as they become intermediate progenitor cells, they delaminate from the layer of cells that line the ventricle and begin to move outward in a radial direction, which, this is a little bit small. I hope you guys can see this. But it's almost like the radioglial signature, but it's starting to move out and sort of looks a little diffuse as these cells are moving away from the ventricle. And depending on how the sort of tissue slice is caught, you can kind of see sometimes on the ventricle, right along the ventricle, it's actually lighter where these cells are moving out. And then we're going to keep moving to the right here now. The newborn neuron signature is nearly devoid or nearly gone all over here on the lining of the ventricle where the stem cells are, because this is not expressed in stem cells. Where is it expressed? Further out radially as these cells have moved through the IPC state, and they are now becoming neurons, and they're moving further out where they will come to rest after that migration in their layer of interest, or their target layer of the neocortex as the developing cortical plate is formed. But you can see the difference between this nascent neuronal signature and the maturing neuronal signature, because this again is at the surface, but it is just at the last edge of the surface. This is the furthest out these new neurons have migrated. They are now coming to their resting place where they will stay for the rest of our lives. And that's when they turn on this mature neuronal signature. And again, here we have brought a whole spatial landscape to the data or the decomposition that signatures, all those signatures we got from single cell data where we had no spatial information. Now we're going to move down each one of these data sets and kind of get the same thing. Information that we did not have in those initial experiments, but we can infer by mapping the signatures. We have an emergency. So here, no. In this next one, in the second row, we don't have space. It is another single cell sequencing experiment, but it has an incredibly cool temporal element to it. Without telling you how, they labeled cells that were dividing on the ventricular surface at time 0. And then when they harvest cells at 1, 24, and 96 hours later, they not only sequence them and have their RNA profile, but they know the number of hours ago that those cells went through their terminal division to become a postmitotic neuron. So in the StereoSeq experiment we looked at a second ago, we're bringing spatial information to these neurogenic trajectories. Now we're bringing temporal information, again, that we didn't have. We just had the age of the animal in those other experiments. And here we're about the same ages. They have E12, 13, 14, and 15. But on top of knowing how old the animal is, we know how many hours ago that cell last divided to become a neuron. And therefore, we can see how many hours it takes in a mouse to move through this temporal progression of neuronal transcriptomic development. So at 1 hour, that cell that divided has high levels of the radioglial signature. So this makes sense. An hour after you've divided for the last time, you still look more like a neural progenitor than the neuron that you are becoming. And then at 24 hours and 96 hours, that neural stem cell signature has come down. But what starts to come up is all the other signatures. So immediate progenitor cell signature is actually sort of moderately high at both 1 and 24 hours. And by 4 days or 96 hours, it's off. And sort of reciprocally, at 1 hour, the newborn neuron signature has not yet come on, but it comes on at 24 and it's already turning off by 96. And again, in kind of a seesaw here, at 24 hours, the maturing signature has not yet come on in the newborn neurons, but by 4 days later, it is coming on. And again, harnessing new data sets to see things that we already saw in other data sets. And the wealth of data out there is really insane. So we can do incredible things when we get these data sets together. And then just two more examples, and then we can discuss any of this. I think we'll have a little time. So this is now adult data from a 30-year-old postmortem human brain. This is, again, spatial transcriptomics. And on the right side, we have this legend where this bottom corner is the white matter, and then we have the differing layers of the cortex moving outward here. So this is a very different context. Even though this is still neocortex, this is now 30 years later because we got our signatures from in utero data. So the stem cell signature is high in the white matter. Now this makes total sense because we know that both astrocytes and oligodendrocyte precursor cells maintain some of the expression modules of the neural precursors. So even though there are no neural precursors left in the adult brain, the concentration of glial cells in the white matter show a signal there. The IPCs show almost no signal, maybe some slight enrichment, again, in these glial cells. The newborn neuron signature is totally gone. There's basically static across this whole thing. Maybe you could argue there's a little bit more in the cortical ribbon and not in the white matter, but really this is pretty flat. And where we really see a signature that has been maintained is this maturing neuronal signature. So something that turned on within the first 4 days of this neuron's birth is still on. It's really just part of being a neuron. And it is not layer specific. This is a pan-neuronal signature that in our 30-year-old brains is still on. And this is sort of just the business of being a neuron. Synaptic genes, axonal maintenance, etc. Okay, and now the last example. So this is even a bigger jump. This is human cells again, but not brain cells. These are modeling brain cells in a dish from human pluripotent stem cells. So this Canton et al. Study in 2019 was among the first that really got some high resolution single cell data out of organoids. So I'm guessing a lot of you know what these are, but I'll give you a 2-second introduction. We can take old adult cells from anybody's skin or blood, reprogram them in 7 days using a protocol that won the 2012 Nobel Prize to convince those cells that they are the first cells, that they are embryonic stem cells, and it's their mission to make the human body. If you nudge them in the right way with morphogens we know about from developmental biology, you can get those cells to think their job is to make the brain. And with well worked out protocols these days, you can make a ball of cells about the size of a pea that has an incredible similarity to their developing mammalian brain. So this is a single cell sequencing experiment from one of these pluripotent stem cell derived systems. So now we can ask the question, how, excuse me, how well are these in vitro systems modeling the in vivo neural development? So here's sort of our key as to what is what. These are pluripotent stem cells back here. The neural stem cells are actually up here. So that's why there's a high radioglial signature there. These are the cells that are modeling those in vivo radioglial cells. You'll notice, though, that this signature is high all the way through pluripotency, which is interesting. Here, in the next step of IPCs, right beyond those radioglial cells, as this trajectory continues, the IPCs are also well modeled in this system. They're turning on right there. And here, right after that, where we're getting neurons, the nascent neuronal signature that we all know lasts for a very short period of time in vivo development is turned on in the neurons of the organoid. And finally, our more mature signature is right out here at the tips, just the most mature cells in that organoid. Now, one piece of sort of really interesting dynamics here is that in vivo, the nascent signature comes up and then it comes down. And we can look at that in hundreds of other data sets. In vivo, that always happens. This nascent neuronal signature lasts a very short period of time. It gets turned on in every neuron that is born. and gets turned off in every neuron that is born. But it is clear, I'll get rid of some of these drawings so we can see, it is clear, so it usually gets, the nascent signature gets turned off where the maturing signature is greatest. And in fact, let me rewind to show you that. So here is our maturing signature in the mouse. It's really heavy here and it's light here. That's right where that maturing signature comes on. In the monkey, it's the same. At the edges is where the mature signature is, and that's just where it starts to get light. Again, in the human, maturing is down here. Before it was high, it's down here. And where it's down is where that maturing signature really comes on. So in the organoid, where the mature signature is coming on, we can say that's a success in recapitulating that element of in vivo development. But what is lacking is you do not see the descent of the newborn signature. So these neurons in this organoid are actually kind of caught in a extremely early state. And we would never have been able to know that by looking at them. And people that look at these organoids, they see, of course, all these mature neuronal markers coming on, and they stain with them, and they say, wow, this is great. We have generated neurons in vitro in these models. And it's hugely opening what we can look at for disease modeling and therapeutic development in the CNS. But once we get into all these decompositions and high resolution projection, we can actually pick apart details that are not exactly like in vivo development. So it looks like they're getting the cell types of interest, but they're not sort of refining them. The things that actually should be turning off in these more mature neurons are not turning off. And I think that is my last slide, unless I've tricked myself. So like I said, I'll try to shoot out an email with lots of links to these data sets, and hopefully it can be interesting to you. And again, even if you have no multi-omics data, you can look at individual genes. Reach out to me if you want to get fancier and look at gene signatures in the same environment. Because we are ready. These, like I said, it took many, many years to get these data sets ready and these tools ready. And we've built them, and now they just need to be put to use. So please do reach out if you think they can be useful to you. Thank you so much.
Thank you very much. It's amazing to see this collaborative work with such immense potential. We have time for a couple of questions. You can either unmute or send them over the chat, and I will read them. Yep, Dr. McGrath.
Carlo, that was a tour de force. It's fabulous. And my question is less about the utility of this and how you manage the collaboration. And I'm thinking from human data, especially when it's multi-omic, you've got genomics, you've got proteomics, you've got transcriptomics, you've got phenotypic characterization. You know, we have a data trust on the human side that manages all of that and make sure that we maintain patient identification. So on this side, you know, obviously it's not majority is not dealing with human data. How do you sort of maintain the collaborations and the collaborative spirit when you're combining these data sources?
Yeah, so as you saw, right, any one page or collection has a huge amount of resources. So a couple things come into play. One is that we buy ourselves out of the patient identification problem somewhat because we are using processed data, right? The raw data that all these data sets come from has sequence in it that can basically is a blueprint for patient identification. So we don't work with any of that. There are also large efforts to integrate these data sets at that raw data level. So you have to be a little more careful, but all the data we have is at the gene level and it's de-identified. So it is, we can trade it more freely. And what that allows us to do is each one of those boxes on that website that allows us to look at the expression of either one gene or a signature in that data set, right with that box. In fact, here, why don't I, so for instance, here, on this pluripotent stem cell data set, if we try to keep as connected as possible to all of the original data generators for many reasons. So if we click here, you can go directly to the publication or the repository where the raw and processed data is. And the other thing you can do, if you just hit the information, I don't know why there's not more data there, but usually not only can you see the paper, but if they have a repository, or often people build a one data set website where you can really dig into that one data set deeply. We try to maintain that sort of web of connection. So whether it's a programmer who wants to go get the raw sequence reads and really get into some splicing question for this data set, or if it's someone who has no bioinformatics skills and wants to explore this data set deeper, maybe with the website that the original publication generated, they can link out to that right from here. So we're trying to do as much as we can in this environment and at the same time stay connected to as much of the community who produced it as possible, if that answers your question.
Any other questions? And in the meantime, you're getting a lot of gratitude in the chat, appropriately so, for what you're doing and how generously collaborative it is. And some new Nemo accounts from Dr. Werner and Dr. Kornberg.
Awesome.
So,
Alex, I see your hand up.
Hi, Carlo. Great talk as always.
Hey, how you doing, Alex?
I'm doing great, thanks. Nemo's gotten so powerful. I feel like there's, it can almost be a little overwhelming when you first open it, even for us in sort of the genomics field.
Yeah.
How would you sort of suggest that a totally new person looking to try it out first interface with it?
That is a great question, and you're absolutely right. Just walking in the front door, there's so much data and so many ways to look at it, it can be difficult. So a couple things. We have a publication on bioRxiv currently under review on the neocortical aspects of this. And here also, I'll pull up a page. We have a landing page for that in particular. But to tell you the truth, I think at this point, for what I presented today, my own website is pretty good. So this is the stem cell environment, the neocortical environment, and the neuroinflammatory environment. And we're also working on peripheral immune here. So this is all on my own website, but links to the resources we have. For instance, if you click on the neocortical development one here, this gives you a little bit of an idea. This is the picture we just looked at together. And one thing is here, this is really a page that is on the Nemo Analytics website that is meant to be a landing page for the neocortical development project. This summarizes the crazy amount of data we have in here. And down here, it mentions the different collections we have for this. And this is where you really sort of need to learn a little bit about what's there. These collections can be big, they can be small, it can be 5 or 10 data sets. Some of these are up to 50 data sets, but we've broken them. Here we actually have a live version of the figures, the first couple figures, but then after that we have a collection of the human in vivo data, the human in vitro data, the mouse in vivo data, and the monkey in vivo data. And then we have a couple other collections that are relevant to cortical layering and so on. But each one of these is a collection of many tens of data sets. And this whole matrix here are links. The row determines what collection. So this, say, would be all links to look at the human in vivo data. The first one is to look at individual genes. And then all the subsequent ones are interesting gene signatures that you might want to look at in that data set. So these are the decomposition I showed you today. The 4 signatures were from a set of 7. So if you want to look at those, you just click on this button. If you want to look at a decomposition that was relevant to the layering of the cortex, it's here. If you just want to look at where the cell cycle genes, and in fact, here, let's look at an example. If I click on this, this is the human in vivo collection looking at cell cycle genes. If we click on this, It'll bring up the collection of in vivo data, and this should look familiar. The whale tails will appear, but instead of looking at a single gene, we've loaded the gene lists that are associated with different phases of the cell cycle. Here, the G2M or the S phase. So it takes a little bit of time to populate, but not too crazy. But here you can see, as we saw earlier, for instance, in this whale tail, where you have your dividing progenitors, this is exactly where this turns up. And that's S phase. So this is almost zooming in more, right, than just dividing. You can actually see which phase of the cell cycle. So if let's outline this so we can remember. Here's where it's hot. That's G2M. And if we now click on S phase, so it moved a little on us, but you can see it moved in, right? The S is here and G2M is here. And of course, you could paint this, it is painted across all of these data sets already. There it is. You can see all the S phase cells in all these cells that were ever published. So the landing page was kind of meant to be, at least for the neocortical development data, what you're asking for, like how do I start looking at all of this? For neuroinflammation, we have kind of one big collection. So it's just a single link. But I'll make sure the email goes out with kind of a listing of these. So if one or another of the environments is interesting to people, you can just go ahead and click on it. And I should also mention, by the way, that if you do have people often who don't do genomics and transcriptomics have a small RNA sequencing experiment. And this system is really built for you to upload your data here, whether it's just for you to explore it or for you to make it available in addition to a repository deposition so that people could explore your data. You could keep it private for a certain amount of time here before you publish it, allowing you to explore it in the context of all these other data sets. And when you publish, you can flip a switch and have your data set be publicly available and explorable right in here with the rest.
Thank you very much. We'll have to move on now, but there are some more questions, including Dr. Green's question on generative AI. I'll ask if you can answer that in the chat. That would be helpful.
Sure.
Thank you again for being here with us today.
Thank you.